So what I am saying that you should have a tabular format in 15.1. Okay. You can also write like this, that the summary of, or the summary of like this new ongoing or closed, the same thing is presented in appendix 3. Okay. And the method that you were asking, so it will be on 15 above this 15.1. Okay. So in the same way you can write, and how that appendix will look, or you can provide a tabular format here also. So how appendix will look, see. Let's say this is the date opened first, signal, then date opened, then
So this date
trigger.
opened is same as date detected, right?
No, I'll tell you. Date open, trigger, and then status, date closed if applicable, and conclusion. Right? This way. Suppose now your signal is suppose thrombocytopenia. You will write here thrombocytopenia. Okay. Date opened. So date opened is basically the date when it is validated. That is your DLP.
Okay, so
Okay.
in this section only validated signal we will take.
Yeah, we have to take only validated signal. So date opened means date when it was validated. So suppose today is, what is today? 5th. 2025. Trigger. Now, what could be the trigger? So suppose a literature, you can say literature or clinical trial, whatever is there, right? From where you find that, from where, or your PMS database, whatever is there. See how I'll write
HA request also.
everything.
Ah.
HA request, whatever source from where we get the trigger for that. Status, suppose ongoing, so you will write ongoing. If closed, then you will write closed. Closed, right? If closed, then date, suppose today's date only we should write here. And conclusion, if closed, then what was the conclusion? Like suppose the RSI section 4.4, 4.4, 4.8 was updated. Okay, this is the simple thing, the tabular
Yes.
format that you will provide. Okay, so this will give a overview of all the signals, whatever is there. Right now, when you are in 16.2, so what will happen? 16.2, what is the signal? What is the topic?
Closed
Heading for
signal.
closed
Evaluation of
signals, whatever is there. So let's keep it as closed signals. Now, closed signals. So many clients used to bifurcate it, that the closed signals which were refuted, closed signals which were confirmed. So I'm just taking how do we present this? No? So it can be subtopics here: closed signals which were refuted, closed signals which were confirmed, or closed signals which became safety under monitoring, whatever it is. So what will happen here? Like you will write trigger / background, anything you can put here. So in this, now you will write that here you have given that literature or clinical trial or PMS. So here you will write what was that literature. So you will write that during the literature routine, literature screening, the MAH identified a literature titled something, whatever is there. Yeah, like this you will write the trigger. Then you will write discussion and conclusion, simple. So suppose discussion and conclusion. So it will be like suppose of the total, I'm just giving an example, okay, of the total 173 cases received from PMS, post-marketing source. Like this, how we write for analysis for all those, okay? And then after writing the discussion and conclusion, you will write, or you can write discussion first and then conclusion. This way also you can, it will be clear ideas. Okay, this will be copy pasted from signal evaluation report. Conclusion done. And this is the difference. Now, can you see what is the difference between
Yeah,
15.1
the
and 16?
16.2 is elaborative actually.
Right, elaborative, but summary only.
Yes,
It is in
summary.
paragraph form, right? Summary only. And now in the last, you will write that the appendix, this signal evaluation report is appended in appendix, whatever is there, 4, 5, 6, whatever, or
So,
maybe 3.1.
yeah. So for each signal, we will use some method for that signal evaluation, right? Like how we are reviewing the data or
That will
something
be
like
there in a signal evaluation report. No need to present
right.
here. Oh.
No, because in that table I saw that there was two columns for one was for method of signal evaluation, other one was for reason for evaluation and summary of key data.
Now that is the signal evaluation report part, no?
No, 15.1 table. I'm saying
No,
like
no,
in
not required. Method of evaluation not required. We can write about the signal detection process. That is a standard template right now.
okay, got it. So
If
like
you
in
want to,
the
like suppose if your client says has a convention to write method of evaluation, then you can write only as to what source you use. Like you use clinical trial source, you preclinical trial source, other labels. These headings only you have to give there. Nothing more than that. No elaborative thing in 15.1.
also Amit, if a signal is identified, like suppose there is a rash, okay, already there is a rash identified in the RSI and
Hmm.
SJS is reported in
Hmm.
some of the reports
Hmm.
within like some period, like 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 there are some 4 to 5 reports came within a short period. So this information will trigger right in trigger background, right?
Please
For
come
that
again.
signal.
What you are saying?
So
Like rash
let's
is
say
there in RSI. Now
yeah,
SJS is reported. So
SJS reported like after authorization SJS is reported
hmm.
within some like 6 months of the period, like there are some 4 to 5 reports of SJS
Hmm.
or some more reports, like it is going to be validated and it is validated.
Hmm.
So we are for the validated SJS signal will
Hmm.
trigger or background will be like this is the background. That's why we are evaluating, right?
Yeah. So like you got those SJS cases, even a single case of SJS will trigger a signal. So suppose if you got those SJS cases in your PMS database, that is Arga. So your trigger will be that during the routine PV monitoring, a case of SJS was identified in the safety database, in the global safety database. Okay.
Right. And okay, yeah. So this total of 173 cases we are bifurcating. So same like non-serious, serious reporter type. Or
The same way we need to bifurcate and find the causality. This main purpose is to find a causality.
right.
Okay.
Okay. Thank you so much.
Welcome. Any other question, people? Shall we move forward today? 17. What is the topic for 17? Tell me, because I don't have open in front of me. Important baseline information. What is that? Important or benefit information? What is there?
Baseline information, something about application.
Baseline. Tell me. Baseline information.
Section 17, na, right?
Main heading.
Main heading is benefit evaluation. I'm looking at the
Benefit evaluation.
that's it. That is the PSUR section I'm looking
Okay.
at.
17.1 baseline, important baseline information at the start of the reporting
Important
period.
baseline efficacy and effective information.
Important baseline.
Efficacy and effectiveness information.
Can anybody tell me what is the difference between efficacy and information effectiveness? Anybody?
So they are similar, right?
Then why we are writing both? There should be some reason why we are writing both. Anybody, any one of you, can you tell me what is the difference between efficacy and effectiveness? See, if there is some heading, then there should be some reason behind it, right? So, and this could be a question to you, like what is the difference between efficacy and effectiveness? Anybody? Yes, no, maybe? So.
Probably efficacy is predetermined and effectiveness is real time. That means how the that means drug
like-
is
Efficacy will be determined in the clinical trial, like, and how much effective it is in the real world. That is the different thing.
Good. So both of you are correct. So actually efficacy, when we are determining certain efficacy, or you can say how effective is the product in a certain indication. So efficacy comes from, means efficacy is related to in the clinical trials. If we are getting the effectiveness of it, or you can say if we are evaluating its indication and it's becoming, what is its efficacy? So that is measured in clinical trials under, you can say, closed environment or closed conditions or controlled conditions. So if you are measuring the efficacy in closed or controlled conditions, like in clinical trials, then it is efficacy. And if it is into the real world or post-marketing, then that efficacy is called as effectiveness. Okay. Both are same terms, but one is in controlled condition and one it is in the real world. Okay. Got it?
So, sir, the main difference is between the conditions, means when it is done,
Correct.
when we check effectiveness in clinical trial, we'll call it as efficacy. And when we see the efficacy of that drug in real world scenario, means like, then we'll call it as effectiveness.
Correct.
Acha.
So from this, what do you understand? That you have to present both the information as to what was your efficacy during the clinical trial and what is that you are observing during post-marketing or real world. Okay. So that's why we say that section 17 onwards is the responsibility of medical affairs or RW team who writes this. But you need to understand, you need to know what goes in this section, because if you will work for a generic company, then you will only have to write all these things. Okay. So first of all, till now, what we studied was related to risk and all those things. Here onwards from 17, means both 17 and 18 sections, they talk about benefit. What is the benefit of the drug due to which you need approval or you want your drug to be in the market, right? So in section 17 and 18, we will discuss about the benefit evaluation and how we are going to present that benefit and evaluate it. Evaluation, there is no such criteria as to how you just present your information as to on what basis you are saying that your drug is effective or is beneficial to the patient being targeted. Okay. So first of all, section 17.1. So tell me, what do you understand by important baseline information? Okay, first of all, one more thing. In this section 17 and 18, you will talk only about the approved indication. Got it? Am I clear?
yes.
You will talk
Yes.
about only the approved indication. No unapproved indication literatures will go here. Strictly, always remember, that this section is for only approved indication. Any doubt, people?
No, sir.
Chalo. Can anybody tell me what is the pivotal clinical trial? Have you heard this term, pivotal clinical trial? Anybody has heard this term, pivotal clinical trial?
Is this related to first clinical trial?
Pretty much related, but let me write this because it is a pivotal clinical trial. Okay. So first clinical trial also you can say, but it is not appropriate to say. What is the definition is that the clinical trial based on which MAH got the approval for the drug in desired indication. This is not a definition from any of the, you can say, guidelines or something. This is what I am writing on my own language so that you can understand it. So what is the pivotal clinical trial? The clinical trial based on which or the data of which you will present and get the indication approval for that molecule. Like suppose if you are going for warfarin for suppose as an anticoagulant, so you could have done some clinical trial for that anticoagulant efficacy check and safety measurement, all those things. So whatever data you will present from that clinical trial to get the approval for this warfarin as an anticoagulant, that trial will be your pivotal clinical trial. Correct? Am I clear, people?
Sir, please explain once again, once like.
So what happens? Like suppose I am testing a medication, a new molecule. Let's say, for example, that suppose tacrolimus, we are testing it for or doing the clinical trial for the first time as an immunosuppressant. So what will happen? You will plan some clinical trial to test it in human beings, right? Are
Yes,
you guys
sir.
with me?
Yes, sir.
Yes.
Yes, sir.
Okay. So that clinical trial, whatever data you will observe, if you found it efficacious in that clinical trial, then the same data you will present for your post-marketing approval to health authority. The data from the same clinical trial that you have conducted for immunosuppressant, tacrolimus as immunosuppressant, right? So that clinical trial for which, based on which you are seeking approval from the HA or health authority for the drug to be used as immunosuppressant in the market. So that clinical trial will become your pivotal clinical trial. Okay? Understood? Not understood?
Sir, in which phase this happens? At what time?
Initially, when we discussed that post completion of phase 3, we file NDA,
Yes.
right? New drug application that we are going for. We need your approval to start the post means marketing phase of this. We want to market the drug. So when you are filing NDA at that time, you will present the data from this clinical trials. Based on this, I am asking for approval of the drug into the market. Am I correct?
Yes.
Am I able to convey what I'm trying to say? Or is there any other confusion with anybody?
Sir, means in short, whatever we submit at the time of NDA, that will go under pivotal clinical trial.
Yes, you can say at that also. But the simple thing you have to understand is that the clinical trial based on which you are asking for approval, you are also correct that the clinical trial data which I am submitting during NDA is the pivotal clinical trial. But in more simpler language, the clinical trial which you conducted and based on which data, based on the data of that clinical trial, you are asking for approval to marketing, that is called the pivotal clinical trial. Because that is the important clinical trial. That is the significant clinical trial based on which I am asking for approval. This is my source, means this is my base or foundation on the basis of which I am asking that my drug is more beneficial. Please grant me approval to market the drug in market.
Got
Understood?
it. Yes, sir. So every clinical trial must be as in pivotal clinical trial because sir हम उसी डेटा को प्रेजेंट करेंगे जिसमें हमारा इफेक्टिवनेस या एफिकेसी अच्छी होगी फॉर द अप्रूवल. So every drug which has been approved should undergo the pivotal clinical trial.
Yes. So if you say that every drug which is approved, it would be having some pivotal clinical trial, this statement is correct. But if you say that every clinical trial which we have submitted is a pivotal clinical trial, it might happen that you won't get approval for that. It might happen that you have done some 8 to 10 clinical trials, but only 1 or 2 clinical trials are there based on which you are asking for approval. So the first statement which you say that if the drug is approved, then the clinical trial data which was presented for in its NDA, that is the pivotal clinical trial. That is correct.
Yes, sir, got
Okay.
it.
But in the
So-
same case, if you come to generic molecules, then generic molecules, you won't find the pivotal clinical trials because they get the approval based on BA/BE studies. So that's why this pivotal clinical trial concept is there in innovator or the life cycle molecule or the MH who has invented that molecule or discovered that molecule and bring to the market.
So Amit, all the clinical trials will not get approved, you are saying?
All the clinical trials will not be efficacious. You won't even present those data in that while seeking approval. You can give information that there was the clinical trial done and this happened to it, but you will seek information approval based on your pivotal clinical trial only.
Where do we see the efficacy for that indication?
Yes, because if you don't present data, then why HA will give you the approval? That's why this section is there, na? Benefit evaluation in PBER and those trial data are presented in CSR and all those things while submitting NDA.
So it does it happen very often, like for a innovator molecule, there are 4 to 5 clinical trials conducted and only one clinical trial is presented?
See, they won't present all this data to you. It happens that they might conduct the same clinical trial on different sites. They might conduct 2 or 3 clinical trials simultaneously. But what happens usually, like suppose my drug could be efficacious in suppose NSCLC also, in Hodgkin's lymphoma also. In that case, I will initiate multiple trials. But suppose my NSCLC trial was there and there were some 2 to 3 trials that I was trying to do. But like one trial I was doing in pediatric patient, one I was doing in adult patients, one I was doing in normal every age group. So what happened that in pediatric, my clinical trial is not getting efficacious. So I'll say that I tried in pediatric, but it was not efficacious. But in the adult patients, it was efficacious. So I'll present the data of adult patients and ask for approval for the that molecule into an NSCLC in adult patients. So this is how the trials happen.
So whatever the data for efficacy we are presenting to HA for as a clinical trial data, so that means it is a pivotal clinical trial. It will
Yes.
get approved, right? Does this happen
That
like
is a pivotal clinical trial for you, but it will get approved or not, that is HA's discretion.
so does it happen like it will not get approved or
Yes.
from HA side?
Yes, it happens that it don't get approved, or it might happen that they would say that initiate some other, they will ask you, means they will give you approval based on certain conditions that you have to do these clinical trials along with this. So these all things happen.
Unless it is not approved, we will not consider it as a pivotal
Pivotal
trial, right?
clinical trial, correct, for that indication.
Sir, the definition in which you have written desired indication. So will formulation be also a parameter here?
Formulation, but if there is some formulation, then it could be for the same indication, right? Or for different indication. How yes? Because this trial is based basically on indication, not on formulation. Formulation is there, then we will seek saying that we need this formulation also approved for this. So that is a different part, but the indication will remain same. So clinical trial is basically done for indication. Any other question?
No.
Tell me, people, any other question? Shall we move forward?
Yes.
That formulation question is clear, na? Who was there? I
Yes,
didn't
it is
see.
clear.
Huh?
Yes, it is clear.
Yeah, because in formulation, what happens now? They mainly look for, like, if I have to apply for some formulation, then and the indication is same, then I will do the BA/BE studies to see whether that formulation is equivalent to what is approved or not. So based on that, I'll get the approval and everything. So in that case, we don't do clinical trials for indication. Indication is same and formulation is different, then we can do BA/BE studies and complete it. Okay.
Okay.
Any other question, people?
No.
Yes, no, maybe.
No.
No,
Sure.
sir.
Then now tell me, what is the important baseline? What do you understand by baseline term here? Efficacy, effectiveness you have seen. Now tell me, what is the baseline information? What do you understand by baseline? Any thoughts? What is the baseline?
So this
Approved
is the
indication.
minimum that is expected from that drug.
Minimal,
Or anything else? Tell me, people, anything else? What do you understand by baseline? Now you people have done
Whatever
so many.
studies which are the starting one means which are not in our reporting period means which are from our previous fever and all.
or initial efficacy means like initial efficacy information. Hello.
Hello.
Hello, am I?
Amit, you are not audible.
No, sir, you are not audible.
You are not audible, Amit.
Hello, can you hear me?
Yes, sir.
Yes, yes.
What is this? Hemant, what are you asking me to do? Smart recording. Decline level. Okay, chalo. Screen is presented, na?
Yes, the screen is visible.
So important baseline efficacy. So what happens? By baseline, whatever we mean is, like, suppose your reporting period for the PBER is 1st January 2024 to 31st December 2024, right? So at 1st of January 2024, so before that means by 31st December 2023, whatever information is available to you on efficacy and effectiveness. That will be your important baseline efficacy. Got it? Means the efficacy and effectiveness information that you have at the start of the reporting period. So that is your baseline efficacy information. Got it? Got it? What is the baseline efficacy information? Whatever information you have till tell me people till
For
Till the
reporting.
start of reporting period.
at the start of the reporting period is your baseline information. Okay. What is this one? New efficacy.
Sir, so means 7.1, it is from IBD to our reporting period, right?
Yes, all this will be from 16.4 onwards. I told you that everything is cumulative. Okay. Are you clear what is the baseline information?
Yes, sir.
Okay. So now if you see 17.2, so this will be what new information related to efficacy or effectiveness you got during the reporting period. That is 1st January 2024 to 31st December 2024. Okay. Is it clear, people?
Yes, sir.
Okay. Now let's come to how do we present the data. So suppose, let's take an example of second PBR. After that, we will discuss about first PBR. First, let's take an example of second PBR. Like, suppose you are writing a second PBR. So what could be your baseline information? That what information will go in 17.1? Any guess? Anything?
Whatever is there in the previous paper.
Yes, of 7.1 plus 7.2.
17.1 plus 17.2 will
Yes.
become 17.1 for your
1.
second PBR, right?
Right.
Now, suppose if you are doing the first PBR, then what will go in 17.1?
TT
Clinical
team
trial.
will provide.
Okay. So the clinical trial data which you have submitted, based on which you got the approval, it may be 1, 2, 3, 4, whatever clinical trials your data you have submitted. So that information will come in 17.1 in first PBR. Okay. What would be the source for that information? What would be the source for that information?
The
Where you will
document.
get that data?
CSR.
Correct. CSR.
Yes, sir.
CSR will be the source, right? For first PBR. And for second PBR, your first PBR will be the source. First source,
So
right?
Amit, this is for completed trials only. We will take the data.
Only completed trials you will submit for approval. No ongoing trials data you will submit for approval. Right?
Right.
Is it correct? Sure. Then what information we present? Information that we present. First. CT I will write now for clinical trials. Clinical trials. Clinical trial. Number. Name. Number or title, you can say simple number and title. Like suppose if you have conducted some clinical trial and its study name was, let's say, WD5432, something like that, and something like that. And then its title could be like, it could be a phase 2, suppose a phase 3 or a phase 2 or a phase 3B or a phase 3A, whatever is there. So you will write that phase, a phase 2 multi-center randomized, double blind, placebo controlled, Like this, you write the name of that, na? Then in patients with whatever title of that protocol is there, so you will mention here. Okay, city number and title. Got it? Got it, people?
Yes, sir.
Now we are discussing as to what information, because if I will write the information here, then it will take whole class to write. Okay. Now,
So
second thing,
this
what
information is CSR में ही रहेगा सब.
everything is there,
Every-
everything will be there in CSR. Okay. Then you will write in short. In short, you will write the method and inclusion, exclusion criteria. Only the important one. Only in just 2 to 3 lines you will write this. And what you will focus more is efficacy data. If it is a clinical trial, efficacy data. If it is a post-marketing data, then effectiveness data, as we learned. But mainly you can say that efficacy data. Efficacy data. So how that efficacy data? So what was your, like, if you are doing some trial for oncology trials, then there will be primary endpoints, secondary endpoints. So whatever data you have related to efficacy, which proves your efficacy, that data will go here, same from the CSR. Okay. If suppose there are more than one trial, then in the same fashion, you will write for all the trials, the data. Okay.
So this is all will be in the section wise in the CSR, right? Efficacy data and all. Section wise will
No,
be
no,
there.
section wise, it won't be there. It will be in a paragraph. Simply, I told you this is the content. So paragraph like introduction, you write now. You don't write like this is mechanism of action, this is molecule introduction, this is formulation. You keep it writing in paragraph. In the same way, here you will write in the same paragraph. Like you can split it into 2 to 3 paragraphs. Like in first paragraph, you told what is the city number, what is the title, then why it was done, what was the method. Then you can write about the efficacy data. Post that in second paragraph, that this was the method used to evaluate the efficacy, like the primary endpoint was decided to be this, and in these many patients or placebo controlled. So whatever data is presented in CSR, in the same fashion you will write. There won't be any bifurcation in the subheadings, like this is efficacy data, this is protocol title, something. No, nothing like that. It will continue in paragraph. Okay. And for first clinical trial, you will cover first. Then for second clinical trial, you will write like in a study. Suppose this is for in a study 1, 2, 3, 4, 5. Then second, when you start, you write in a study 5, 6, 7, 8, 9. This is the same thing. In the same fashion, it will be written. Okay. So if you will see the model paper that you have now, in that you can see, you will find how they have written the, or try to download some, if there is some molecule paper other than that vaccine. So it will be more clear to you. You can find how the data is written. Okay.
So the data from CSR we will have to summarize here.
Yes, you can't write the whole CSR here, right? You will just summarize the main thing, means mainly focusing on efficacy. You have to present the efficacy data here. No need to have providing everything. Whatever data supports your efficacy, that will come here. Okay.
So Amit, can you give the example how efficacy data looks like how many patients were treated and is it like that?
See, I'll tell you how the data looks like. Like suppose, I can't. Yeah, if I will write, then it will take like the data. How it will be there?
Just
Na?
verbally.
Verbally, like suppose if you are writing the study title, you will write. Then you will write what was that? Was a, like suppose a study Feder MF or a study. You can say that some 2369 something like that was what was that study? So it was a multi-center or single center or randomized or open label. How is it? So then was a multi-center, suppose randomized single arm study or double arm study in patients. This is the title that you will get from CSR. After that, you can write why that study was conducted. So it was conducted for in the patients who had a diagnosis of suppose NSCLC. Then you will take how many patients you took or how many subjects were enrolled in that. suppose there were 40 patients or 40 subjects enrolled and who started treatment with your drug, then you will say that what was the age group distribution in that. Suppose 30% were male, 40% were female, like this. Then after that, when you will start explaining the efficacy data, you will say that suppose it was for NSCLC, so there would be some primary efficacy endpoint, there would be some secondary efficacy endpoint. So you will write like the primary efficacy indication for the subjects achieving, suppose you can say what was the efficacy endpoints. Like you keep a efficacy endpoint in NSCLC as to suppose greater than 40% reduction in the suppose that cell volume, something like that, from baseline to the like end of the some cycle, because oncology is there, so there will be some cycles, 1 2 3 4 5 6. So you have targeted that by the end of cycle 8 or 10, your that NSCLC, the primary endpoint, what you have kept, like the cell volume increases to something like that, more than 40%. So are you trying to get like there would be some achievement points or efficacy achievement points on based on which you will say that my drug is more effective. So if those achievement points are how those achievement points are reached, that information you will present here. So everything will be there in CSR. You just have to go, you have to identify what information has to come here. Okay.
So, Amit, for each patient, the efficacy will be different, right? Different, right?
So that's why we write in cumulative. Like suppose if your molecule was exposed to 50% 50 patients, then in that 50 patients, how it happened? Like you were trying to achieve 40% of cell volume increased. So 5 fours are 20. If more than 20 patients have achieved that volume, then you will write that 22%, 23% patients, or 40% or 45% patients have achieved that volume. So it will not be per patient per se. It will be a cumulative data that you will present.
Okay, got it.
Okay. Chalo. .1 is clear now, right? Right, people? Now, let's come to 17.2. What is the new efficacy or effectiveness information? So suppose your drug was approved for NSCLC in certain region like European region. Now you are conducting the clinical trials, the same clinical trials in Japanese patient, or say in Latin American patients, or you say US patients, same indication, or maybe you can say because whatever data you have to present is for the approved indication. So it might happen that some other indication also got approved, but let's take the example of same indication right now, not to confuse you. So suppose, as I mentioned, that it was approved in European region. Now you are doing the clinical trial in Japanese region where you found the efficacy of your drug in Japanese patients also. So that is done during your current reporting period. So that trial data will come in 17.2 as a new efficacy or effectiveness information.
And the same way we will write, right?
Same way, same way. The way will be same. Okay.
And for all the indication we have to write.
For all the approved indication, you have to write. All the approved indication, whatever clinical trials done for them, all information will go here. Okay. Am I clear, people? Any doubts?
No, sir.
Chalo. Then let's go to section 17 point. 17.3 is there. Characterization of benefits. Right? Yes, no, maybe.
Yes,
Yes,
yes.
sir.
So what should go here? Characterization of benefits. What information we will present here? Any idea? This is simple section. You don't have to search anything or write. What you have to do is just give information about molecule. About molecules means it's chemistry, you can say chemistry or class. Okay, like suppose if it is some like imidazole antibiotic or broad spectrum antibiotic or some inhibitor or something like that, that chemistry or class, simply one line, just from the RSI you will take this, whatever mentioned in the RSI. Okay, then what you will give? Whatever information you have presented above in 17.1 and 17.2, what efficacy information you have presented. Okay, so the same information you will give here in 2 to 3 lines from means like suppose you presented 3 trials here. So you will present that the efficacy endpoint, primary efficacy endpoint was reached in all the 3 trials. Like in study 1, 2, 3, 4, it was more than 40%. In study 4, 5, 6, 7, it was more than 35%. In the same way, that's just a single liner. What was the efficacy that you observed? You can provide here as a efficacy data from all trials listed above in 17.1 and 17.2. What efficacy data? Not the whole thing that you are presenting in 17.1 and 17.2, but only the main primary, like primary efficacy endpoint in oncology trials, or there could be some registrar score reduction in DRESS or SGS symptoms. So the main objective which you wanted to achieve from the trials, so that objective will be provided in a line or two, or in a paragraph, you can say, for all the trials. Okay. In a paragraph, not more than that. And based on that, you will give a conclusion that based on comprehensive analysis of assessment of like all new efficacy information during the reporting period, no changes were made to the summary of efficacy information. And this drug has found to be beneficial in this indication. Simple. It is a simple section. Sometimes what happens, generic molecules, so it is always not applicable. But in innovator molecule, there is a standard that you can write from the previous paper. You just have to copy paste. There is no new information. If there is nothing in 17.2, then you just need to copy paste from previous paper. If you are writing first paper, then you can write in the same manner that I have mentioned here. Okay, got it? Any doubt, people? Tell me.
So we can say that this section are written by medical affairs.
Yes, you can say, because most of the innovator molecules, they have a separate team to write this. Either the clinical trial team or medical affairs team will write this. But if you are giving an interview for generic, then they would expect you to write this section. So that's why you better should know as to which company you are applying. If it is an innovator company, then you can say that it is written by medical affairs or clinical trials team, because they are also doing the same thing. And if it is for the generic molecule, then you can write as to how you are presenting this information.
So Amit, how do we know that this company is an innovator or generic?
Like suppose if you are applying for J&J, Pfizer, BMS, Novartis, all these are innovator companies. If you are applying for Apotex, or you can say Sandoz, or you can say Teva or Glenmark, these are all generic companies.
But if we are giving interview for a vendor, then we will not know.
So if you are giving for vendor, then mostly it will be innovator. You can expect it to be innovator by default. They might be that is generic, or they are doing J&J's consumer products, but you can assume it to be a innovator.
So
That's
right
why you
away
are
they ask the question like, do you write this section or do you know about this
so you
section?
said you say that you are working, since you will say that you are working for innovator company. So these sections are no need to mention also that you are working for innovator company. If you will say that these sections are written by medical affairs team, then they will understand that you are working for innovator companies.
Okay.
So one more thing I would like to tell you here, like this is for the innovators what I have explained. For generic, what happens, since they don't have their own clinical trial, right? You guys understand that they are doing a generic, they are the generic company. Like suppose Sandoz is there, so they are having mostly generic molecules. So when they are applying for a generic molecule or you are writing a paper for generic molecule, so 99% it will happen that they have not done clinical trial for those. There was some other innovator for the molecule, and when the patent expired, they have applied for generic MAH and they got it. So in this case, what you will have to do, you will have to search open literature like PubMed and all those things, and you will have to find whatever clinical trial data is there related to the indication for your molecule. You will have to present it here, the summary of that abstract. But you will present in the same manner, like you will write the protocol title, what was the number, means number, it might not be provided. You will write the protocol title, what was the objective that they were trying to reach, and what was the efficacy results. So you will search the open literature and present the relevant literature here. This is the difference when you are writing paper for innovator or generic. Got it? Got it, people? What is the
Yes.
difference between innovator and generic writing of this section? Sure. Now let's move on to section 18. What is section 18? Section 18, tell me.
Benefit risk analysis.
Integrated benefit risk. Bolo, tell me what is this?
For approved indication.
18.1. What is the heading for 18.1? What is the heading for 18.1?
Benefit risk, context, medical and non-important alternatives.
Chalo, now tell me what do you understand by this? Chalo, what do you understand by this? Medical need and important alternatives. What should be the information here?
So why we have conducted this clinical trial for this indication? What is the need of this?
What was the unmet medical need that you are saying you will achieve if this molecule is approved for marketing? And what are the other alternatives? So what do we write here? First of all, mostly open literature is used to present this information. So first of all, like suppose your indication is NSCLC. So you will search the open literature, or if you are for innovator, then CSR, they will be, you can find the information there also. But let's assume that you don't have a CSR and you are searching it from open literature. So NSCLC. So what you will do, you will search about the disease. Like what is the pathology of this disease, or what this disease is. Basically, then you can search the open literature and you can find what is NSCLC and what is its pathology, means what is the pathophysiology of this disease. Okay, about the disease pathophysiology and all, you will write here pathophysiology. Okay, then you will write incidence and prevalence. You will search incidence and prevalence data. Means like what is the incidence of this disease in normal population, general population? How prevalent it is, means how much of population is it affecting? Like suppose if you are writing the PBER for like European region, then you will focus mainly on European region. The region for which the PBER is, if it is a global PBER, then you will present global data. But suppose if you are writing some region specific PBER, then you will focus on that also, apart from global. So globally, let's take an example that we are writing a global PBER. So whatever incidence and prevalence, what do you mean by incidence and prevalence? Like how frequently this disease is found in the general population or in the normal patient, how many patients are getting this disease. Like suppose you have a global data, so it says that in US there were 40% patients, or you say that 2% of the general population gets this data. So this kind of incidence and prevalence data you have to present here. Okay. After that, and focus, if there is some regional people, then you need to focus that you additionally write one paragraph on that region. What was the incidence and prevalence in that region? Okay. And then you will write what are the important alternatives. Important alternatives means like what are the other medications that are being used for this NSCLC other than your molecule. You will not write your molecule data here, other molecules. So how you will present that? You will write the class of the drug, and then drug name. The same way, there will be many drugs that are being used for NSCLC currently. So you will write what was the important alternative for that. Correct? Got it? Correct paper? Did you get it?
Sir, I have a question here. So we are not
Yep.
including a brand name here, right? Just the class of the drug.
Does the class of the drug, like suppose if it is, let's say, just take an example. Like suppose if you say that it is
Immunosuppressant.
chalo, immunosuppressant only. Immunosuppressant. Suppressant. You will write the class like this, and then you will give, like suppose if tacrolimus is there, sirolimus is there, many other drugs are there. So whatever drugs are being used for NSCLC as an immunosuppressant, those only you have to present here. Tacrolimus, sirolimus, etc. You write. Okay.
Thank you.
So this is how we write the important alternative. So section 18.1, this is how we write. Tell me if there is any doubt. No doubt?
Hello, Amit.
Yeah.
Amit, in RMT na
Hmm.
section in the part two in S1, there's
Hmm.
an section like epidemiology of
Yes, you can take incidence
indications and
prevalence. Yes, you can
from
take
there also.
incidence prevalence data from there,
Okay, okay, yeah.
because that is the incidence prevalence means epidemiology only, and that is provided mainly by the RWE team. So you can take data from there. Otherwise, you can open search. If there is no team providing, then you can open search. You can take from RMP. It might be there in CSR also if you find. So there are different sources that you can use.
Same for the about the
Okay.
disease and pathophysiology, right?
For this, for which one?
About the disease and pathophysiology.
Disease pathophysiology will always will also be there in RMP and CSR and everything. In protocols also you can find. But in case if you are not having those data, then you can search from open source. I just discuss open source because it is mostly people will ask as to if there is no such source available, then how will you search this data? So you can say we search from open literature and present the data in this form. Okay. Any other doubts? Any other doubt, people? No. So what is section 18.2 now? Tell me. Tell me, what is 18.2?
Benefit risk evaluation.
Correct. Benefit. Chalo, what information will go here? Now tell me. You have been studying so long periodic benefit risk evaluation report, periodic benefit risk evaluation. Piber's name is right, this only, right? This is the main name of Piber, periodic benefit risk evaluation report, right? So what information should go here? Tell me. Tell me, people, what information you will present here.
So we will present like.
Will there be some method to evaluate this benefit risk? Will there be some formula or method to evaluate this? Yes, no, maybe. Tell me what is your thought process on this.
There must be some
We
method,
can discuss
sir.
further on that.
There must be
There
some
must be
method
some
by
method.
which we can.
Tell.
There must be some method to identify the benefit risk evaluation, I think so.
Okay. So ideally, there is no such method or formula to identify this. In this section, what we present is, see, first we will present like your molecule. What information I should write? Information present here. Okay. So first we will provide information about molecule. Means molecule intro, you can say intro, like class, MOA. This is the main information that go for molecule intro. Okay. Now, after that, you will present the risk or safety information. Like, suppose if you say that whatever in section 16.1 you have presented now, so you will write, like, suppose your molecule A summary of safety concerns. No? So molecule A, like molecule A, or you can write like this. What should be the statement? As the safety profile, which includes important identified risks of blah blah blah, whatever is there, and important potential risks of whatever is there. Okay. After that, what you will do? This is there. Okay. Now, suppose if there was some signal confirmed during this, right? Because we are writing about safety right now. So suppose if Agni signal is closed and confirmed during the reporting periods. So present info in short. Short means like you will present that this signal was identified and confirmed as a important potential risk during the current reporting period. So you will present the information like this. Then once the safety information is completed, then you will write another paragraph for efficacy information. Efficacy information. So what efficacy information? That whatever clinical studies during a reporting period, if you have found some new efficacy information, you will present it here. If there is no, then you will write that there is no change in the efficacy information from the previous reporting period. In the same manner, you can write. And then finally, the conclusion paragraph. Conclusion paragraph, mostly it will say that based on the evaluation of the safety and efficacy profile of your molecule, it is found to be beneficial in the patients of NSCLC. Hence, there is no change in benefit risk ratio of benefit risk of the information, the benefit risk of the patients for NSCLC. Got this? How do we present this data in benefit risk evaluation? What data we need to present? Mainly, first we will write what was the molecule class and mechanism of action. Then we will write whatever safety information. It is from the above only. From 16.1, you will take that identified important potential risk information and you will present here. If any signal was closed, then you can take from 16.2 and you write that there is what was that signal, how means just in 3 to 4 paragraphs, not like 16.2. You will just write what happened to that signal and is there any change in safety due to that. Means is there any change impact on benefit risk? No, nothing like that. Then you will write the efficacy information, whatever efficacy information you have found till in the current reporting. If any new information you can present here, otherwise you will write that all the clinical trials that were conducted has proved to be efficacious for the patients being treated for NSCLC and all those things. And then you will write the final conclusion. That is the standard template that you will for conclusion is there. So you will write based on the current available information, there is no change in benefit risk profile of the molecule for the patients of NSCLC. So this is how the 18.2 section will conclude. Any doubt till here?
So this will be the repetition of information, means like.
But you are giving in one paragraph as to what you have concluded from the whole paper that you have written. Okay.
Sir,
because
but
this
how will
is
it conclude, sir? Means
Huh?
till 4 we can get it, means like we are copy pasting from all the above sections. Like if we from conclusion paragraph, I didn't understand that how will we how we will conclude, means like.
No, this is the conclusion paragraph for 18.2. This is not the conclusion for PBER. Okay. This is the section conclusion paragraph for 18.2 that based on the information, see how you are concluding, like you evaluated the safety parameters, whatever you had, like all the risk and everything. You didn't find any new information, right? You presented the benefit information, all clinical trial efficacy data or post-marketing effectiveness data, everything. And can you please mute? Who is there? So now you have presented the safety information. Now you have presented the benefit section 17 onwards. You have presented the efficacy information. Everything is there. If efficacy information, something is adding up also, then also it is beneficial for you. There is no change in benefit risk impact. Means the risk is not changing, so that you can say that there is some change in benefit risk from the previous paper. So that's why we are concluding with the standard statement that based on the information or based on the analysis of all the safety and benefit information, there is no change in the current benefit risk profile of the molecule, and we are giving the standard. Don't think that there will how you are writing, means you have presented everything in all the above sections based on which you are writing these conclusions. So what do you have any other doubt?
No, sir. No, sir.
Okay, because this is not where we are going to change the benefit risk profile. It won't happen. If you will write the PBER, the 99.99% or 100% PBER will go like this only. There will be no change in benefit risk profile, because otherwise your molecule will be out of the market, right? So most of the PBER, it will be the same. Somebody might confuse you saying that, did you write any conclusion in which there was change in the benefit? You can straight away say that no, till now I haven't faced any such situation. Okay.
So, Amit sir, what all information will be included for safety information? Because in second point, there is risk or safety information, both the
So
things are.
safety information, I told you, na, risk of safety information like important identified risk in section 16.1, what you are presented. Important potential is no missing information. Only important identified and important potential risk. And if any signal was closed and confirmed during your reporting period. So you can present that.
That is for risk and safety, for safety information.
The risk is the safety information. The risk is only the safety information that you are presenting in PBER.
Okay.
Right?
Mhm.
And then 17 onwards, whatever efficacy information is there. Okay.
Amit, in 17th section, we mentioned efficacy in the clinical trial. What about the effectiveness in real world data? How we will present in 17th section?
Like suppose if there is some other, you can say that there would be some other clinical trials ongoing for the same molecule, or there would be some other, means there would be some investigator sponsored clinical trials ongoing for that molecule. So whatever information is there from the post marketing, suppose some literature, like suppose your molecule is approved into the market. Now somebody has taken your molecule and they have started some trial, or you can say some testing in there, and they have published an article on that. So whatever article they have published, if that is relevant to your efficacy information for NSCLC, then you will take that article and present it here in the section 17.2. Like if it is during your reporting period, you will present in section 17.2, and you will say that efficacy endpoint and everything was reached there also. Got it?
So whatever pharmacovigilance activity we are doing, those are for safety, not for efficacy. So we will not take account for those
No,
reports.
whatever PV activities you are doing, those are for safety. Got it?
Yeah.
Chalo, then let's come to last section, that is 19. Or you can say, what is the last section? Right? So what do you think? What information will give?
Again, benefit risk.
But it will not present to you like important identified risk, important potential risk. Just it will be like, you can say that based on one, I can tell you, like based on the comprehensive analysis of safety and efficacy information received during the reporting period for your molecule. Simply it goes like did not reveal any new or significant safety or efficacy concern that change the current known positive benefit risk balance of your molecule for the treatment of the patients with NSCLC, like in this example. And then if there is any new signal, as I mentioned, so if there is any new signal, you can write that during the reporting period. A new safety signal of this was evaluated and closed, due to which the CCDS was updated or whatever was done, because it is confirmed, only confirmed signal we are writing here, not the refuted one. So, and then in second paragraph, you can write that the current known benefit risk balance of your molecule remains positive for the treatment of the patients with this. And then the last paragraph, the company will company will continue to monitor the AE reports as well as the identified and potential risks of your molecule by means of routine and any proposed additional PV activities or RMMs. Simply, this is the conclusion. This is nothing much that we write in conclusion.
In which section we will write about the safety issues under monitoring in this section only?
No, 15.2.
No, like if you want to
Conclusion.
conclude.
If there is any safety issues under monitoring, you can write here that the company will continue to monitor. I am saying the last paragraph that the company will continue to monitor the AE reports through routine or additional PV activities related to the important identified and potential risks and safety issues under monitoring. Simple. You will write like this. No need to mention what is important identified risk for your molecule, what is important. Just you will write a simple blanket statement that the company will continue to monitor all this. Okay. Okay, people.
Yes, sir.
Yes.
Any doubt till here? Tell me. I should close the recording because there is some limit for the recordings. Check. Shall I close the recording now? There is nothing like that.
Yes, sir, you can.
No doubt till here, right? So I'll save it. I'll send you this just now so that अरे screen paused. Recording on करते हैं. My screen is visible, right?
Yes.
Yes, sir.
Let's complete the executive summary. Just a minute, people, huh? Hello.
Yes, sir.
My screen is visible, right?
Yes.
Oh, executive summary. Now, executive summary is the standalone document, okay? Why this summary is written? Because sometimes it happens that the MAH would say that just send me the executive summary, no need to send PBER right now. So they sometimes they want the executive summary. So that's why executive summary is a compilation from the below only. There is nothing new written in that this section. Okay. So what you will do, you will just you have to copy paste from the below whatever you have mentioned in the PBER. So from introduction section, you will present like information that goes here. So first you will write the same thing like this is the first PBER and all those things, na?
Are you writing something with Amit?
Yes, can you see now?
The screen is visible, but we are not able to see anything apart from conclusion and actions.
I guess
Okay.
it gets stuck.
No, sharing is paused. Can you see now the screen?
No.
No.
Screen is visible or not? No.
No, no.
No.
Is it visible now?
Yes.
So executive summary, see information that goes here, right? Shit. चलो ठीक है. You will write the first paragraph of your introduction like a report number, DLP, IBD, reporting period, whatever you write in the first and second paragraph of your introduction. Okay. Then you will write molecule, about molecule, mechanism of action. Okay. And then formulation, indication कौन है छोड़ दो formulation, all these things you will write just in short, and then you will write about section 2. You will take worldwide MAH status. Just you need to copy paste from below, not everything, just like worldwide MAH status. So you will not write everything. You will write like this molecule is approved in so and so many countries, only that much, not everything. Then from RSI, you will provide action taken during safety reasons. If there is any action taken from all the sections, you just have to write all those things. Okay. During action taken during safety reasons. During action taken during reporting period. For safety reasons. Okay. So in this section, what will happen? If there is any action taken, then you will write it here. Otherwise, no need to mention also that there was no action taken during safety reasons. Then for RSI information, what current RSI you are using. If there is any change, then change in short, not like you are presenting in section 4. After that, exposure data. Exposure data. Just the final exposure number. No method nothing from clinical trials and post-marketing. Like you will present so many patients were exposed during clinical trials for placebo for your molecule. And then post-marketing, what was the cumulative exposure? What was the interval exposure, reporting interval exposure? Simple data, just number, nothing like apart from that. Then after this, literature, all those things, nothing. If there is any new signal ongoing, new signal closed, new signal closed and confirmed, that you will present here. Otherwise, no need to present. New signal closed and confirmed. Then your information about all the important identified and important potential is important identified, potential, and missing information. Everything will just the enlist. You will enlist that these are the important identified, these are potential, these are the missing information. If any new information found, means if any new significant information found for any of the risks, then you can present here. Otherwise, you will say that there was no new safety information, no new significant safety information found for this. And for then you will go to directly section 18.2. Important benefit risk analysis section. You will present the conclusion from there. Like you will take important identifier you have presented and you will present the conclusion from section 18.2. If there is any new benefit information in 17.2, you can present here. New benefit information or efficacy information during reporting interval, you can present here. And then conclusion. Conclusion will be combined of 18.2 and the final conclusion 19 that you are presenting. So you will present in the same manner. So this is how the executive summary is drafted. Okay. Got it? It is just a copy paste. You don't do anything. Got it, people?
Yes, sir.
Now I will share this. In executive summary, we don't write anything new. Everything is from the below sections. It is copy pasted from below sections. Okay. Stop share. Any questions you can ask me? Now the record.
So